right, so far in our discussion of linkage, we've looked at some definitions about linkage, explained what it was, that you've got two or more loci, or many thousands of loci carried on a single chromosome at a time, but right now we're just concentrating on two. We use the kind of the extreme cases of independent assortment versus complete linkage in the last lecture part. Now we're going to move into the middle and talk about incomplete linkage. So let's take an example of that. Okay, here's a little problem set up. You've got a couple of mutations in fruit flies with cinnabar and vestigial. These are both vestigial wings or short stubby little wings. Cinnabar is a brownish eye color that's different from the red pumice colored eye that you would see. These are both recessive, so we're dealing with a simple dominant recessive system. Again, nothing fancy at this point. And we set up a cross, and the cross is a heterozygous normal. Okay, so dihybrid het, because we're dealing with two traits, right? Eye color and part, and we're dealing with the other one being the wing shape. So we're dealing with these two different ones. And it's test crossed. Remember, test crossing or back crossing is to the recessive, right? So that's just a terminology thing. So when we do that, we get this outcome. We get some normal normals, and we get some normal vestigials, cinnabar normals, and cinnabar vestigials. All right, so what would we expect to be going on? That becomes the question that we might think. Well, we're crossing a normal, all right, with a cinnabar vestigial. We're told that normal or the regular colored eyes, red eyes, is wild type, and it's dominant over cinnabar. And normal wing shape is dominant over these vestigial or small wings. So we're told that this is a heterozygous individual, which means it's a dihybrid het. It's heterozygous at both. This is a complete recessive. And so we've got a simple little Mendelian outcome, right? We're crossing a het, big A, little a, times little a, right? If you want to, once again, go back to your little cross over here, just because we're changing the letters, don't get all confused. We're still crossing something where we've got a big A, little a, times little a, something like that, all right? It's true for both loci. And the outcome, as we would expect, to be fairly straightforward. It would be, our expectation is that we would get one normal for every one normal vestigial, for every one cinnabar normal for one cinnabar vestigial. So 1 to 1 to 1 to 1. Simple cross, right? Well, obviously we didn't get that, right? That's not 1 to 1 to 1 to 1, because it looks like that probably adds up pretty close to 1,000. So we expect, what, about 250 to be this, 250 to be that, 250, something like that. If we did a chi-squared test, it's going to be in the hundreds as far as our observed chi-square. We did not get a 1 to 1 to 1 to 1. But interestingly, if we just follow Mendel, Mendel says we can still separate out the eye colors from the wing shape. We've got some normal and some cinnabar here. I wonder if they actually fit a 1 to 1 of our segregation ratios. Well, there's a 490 to 510 if we count up how many normals we have and versus how many cindabars we have. And in fact, it fits nicely fits a 1 to 1, exactly what we expect from Mendel's first principle. How about for wing shape? These normal wings versus these little stubby wings. It looks like a 500. That's a perfect 1 to 1. All right. So what's happened here? What's happened here is linkage. And I said linkage, okay, as we go on, we think about what's going on with linkage, right? Mendel's first principle applies. Segregation applies, right? We separate these homologs from each other, which is the principle of segregation as we go through meiosis. What fails to happen now is we don't have these things on separate chromosomes, on separate homologal units. We have them together, carried, and they don't separate. So Mendel's first principle is fine. What we're doing is we're violating Mendel's second principle. We're not able to put these segregation ratios back together in order to create this 1 to 1 to 1 to 1. It doesn't, it's not what we expect to see in these cases because we have linkage now, right? So we're violating Mendel's second principle. Something important comes up on test a lot. Now, this is a case of incomplete linkage, okay? Incomplete linkage is where some recombination, therefore some crossover occurs, and the amount of recombination is strictly dependent on how far the two loci are apart, at least for our purposes, okay? It turns out in the real world, in very complicated systems, recombination depends on a number of things, but for us, we're just going to go with a nice linear relationship. If they're really close together, you don't get much recombination. If they get really far apart, you get lots of recombination, okay? So here we are. We've got independent assortment. They're on separate chromosomes. They don't care about each other. No big deal. complete linkage, they're completely linked, right? We don't get any separation. But now we're somewhere in the middle. Incomplete linkage falls between independent assortment and complete linkage. If they're very, very, very close together, then incomplete linkage goes almost to complete, right? We don't see any. If they're very far apart, in fact, with two loci, if they're more than 50 map units apart, and you can test why that's true, they fall back here toward independent. So we're somewhere in the middle here with incomplete at all times. Okay. Now, just to show you what's going on here, we've got this, no crossover would be complete linkage. You can't break those apart. Crossing over and recombination allows, and again, it always shows for the middle for some reason, but again, it could be any of these. But what you end up with, what's important for us is that if you have recombination, you're going to end up with one of these sister chromatids, one of the four of the tetrads that's going to be non-recombinant, like the original parent. We call that parental-like. There'll be another parental-like non-recombinant, and then there'll be two recombinants, as we call. The proportion of these recombinants to the non-recombinants will depend on how far A and B are apart. Very straightforward relationship. Okay. All right, so let's take a look at this. Go back to our simple cross. We are changing the crosses. We're just going to go with this simple little cross, right, which is exactly what the problem said. Under Mendel, we expect a 1 to 1 to 1 to 1. No big deal, okay? Now, let's think about now, with complete linkage, we didn't care, there's just no crossover, but now we're going to have crossover and recombination. That depends on the distance. So let's make up a distance. It'll make it just kind of easy for math, okay? We'll just say the distance between A and B are 20 map units. That means that 20% of the time, there'll be a crossover between the A and B locus. That's what it means, right? At least two of the chromatids will cross over. All right. If it were 40 map units, these would be further apart. And it'd be 40% of the time you could cross over. If 2 map units, they'd be really close together, and only 2% of the time would you get crossover. That's all it means. Nothing magical about this number. Some students look at this thing and this talk and just fixate on 20. I'm just picking 20 because it makes the math easy, okay? You can put 32, 49, 28.7. I don't care what you pick, but that's just a simple one for us, okay? All right, so with linkage, what are you going to get, right? Well, with linkage, because of this recombination represents 20% of the time, you're going to get something that looks maybe like this, okay? You're going to get all 4 genotypes, but they're not going to be in a 1 to 1 to 1 to 1, nor are they going to be in a 1, 0, 0, 1. They're going to represent the amount of time that you get recombination. Okay. Now, where the 10% go and the 40% go depend on this particular genotype and its configuration. So we got to go back to that discussion about configuration, right? Let's take this genotype, it's one we're working with. Again, it could be set up in a coupled configuration like this. And if for those that are non-recombinants, we know we're going to get big A and big B traveling together, little a and little b traveling together. We're going to get those gametes, right? But now, because the other ones, and we're drawing them in the center just for simplification, for this is a cartoon anyway, because these can cross over and exchange information, have recombination, okay, we can actually end up with this. We can actually end up with big A crossing over and picking up little b, and little a crossing over and picking up big B. So we should be able now to get these gametes, right? Okay, but they're not going to occur in the frequency of 1/4, 1/4, 1/4, 1/4, right? Because this is not going to happen as often as not anything happening, okay? These outer ones are the started away, the configuration started are called the parental-like or the non-recombinants, and these are called the recombinant gametes because they recombined their form, okay? All right. Let's take our study where the two are 20 map units apart. Again, if you want to use 30 or 40, it doesn't matter. It won't make a difference, okay, except it'll just change the numbers, okay? In that case, 20% of the time, you're going to get recombination. Subtracting that from 100%, 80% you must not going to be, okay? That's going to be the parental-like. So 20% are going to be recombinants, you're going to get these, and then 80% of the time you're going to get these based on that map units, right? So let's set this back up in a coupled configuration, something like this, all right? Linkage, all right? Again, this side's basically irrelevant. Okay, don't worry about it too much. It's just a placekeeper. It's this, everything is happening in this genotype over here. All right. If we were following pure Mendel, we'd expect 1 to 1 to 1 to 1. If we were following a coupled configuration with complete linkage, say male fruit flies, we'd expect 1, 0, 0, 1, right? Half of them would be one type, the other half would be the other form, because we'd only get these gametes right here. We wouldn't get these. But because we're in an incomplete linkage, we're getting some of these and some of these, right? So the outcome from that looks like this. We get about 40% those, 10, 10. Where did I get the 10s and 40s? 80% of the gametes are like this. There's 2 possible gametes, so divide by 2, so that's about 40 and 40. And then 20% are off here, right, representing these 2 gametes. Okay, so see where we're coming from. This is big A, little b, right? Big A, little b. These big little a's and little b's, you don't worry about them because they come from here, right? So 20 divided by that. It's that simple that we would get that answer, okay? Now, what happens if we change repulsed? Watch what happens here, okay? This is repulsed. All the repulsed did is that now this big A, little b came down from one parent. That came from over here as big A, little b. And from this parent, it came down as little a, big B, okay? Instead of big A, big B coming here. That means nothing changes except the gametes proportions. Non-crossover, the parental-like, is going to be the most common things at 80%. And recombining, which puts big A with big B and little a with little b, is only going to happen about 20%. Okay? So under linkage, again, under strict Mendelian, we get all 4 of these in equal numbers. We expect about 1 to 1 to 1 to 1. In complete linkage, because we got repulsed, we expect none of these and none of these, but 50% of each of those. But because we've got this linkage problem going on, we expect to see something that looks like this, right? Now notice all we did was flip these around, okay? We didn't change anything. It's just configuration determines which one of these gamete pairs is going to be the largest and which one is going to be the smallest. All right, so let's go back to our original question. What's going on here? Well, the first question, and there are almost always 3 questions that come up when you start working these linkage problems, particularly the 2-point mapping problems where you have 2 loci. First is, are they linked? Well, depending on the cross you made, it's almost always one of these little test crosses or recessive back crosses. You expect a 1 to 1 to 1 to 1. If in fact you violate that, okay, then yeah, they're linked. That's by definition, they're linked, right? The reason for that you can know that is you violated Mendel's second principle, the principle of independent assortment. You have not violated Mendel's first principle, which is segregation, right? But you have violated the second principle. Okay, if they are linked, then you'll always be asked, what's the configuration? Well, here we've got the dominants traveling together and the recessives traveling together. That would sound to be coupled, right? You can try that out, but it's coupled configuration. So this is coupled, okay? And so we've got normal, normal, normal vestigial, and we've got a coupled configuration because the pluses, the wild types are traveling together, and the recessives and the cinder bar and the vestigials are traveling together. And this side over here is a moot, okay? All right, so that. Now, if they're linked, okay, you always ask, what's the configuration? Problems always work with that. And then we're interested in the distance. The distance being, we can use this information to tell us approximately how far the V locus, the locus that's controlling vestigial wing, is from the eye color locus, the cinder bar locus, okay, the eye color locus. You do that because remember, 1 map unit is equal to 1% recombination, right? Well, here are our recombinants, and we got a total here, okay? Happens to be an easy total. Looks like 35 plus 45 is, out of 1,000, it gives me 0.08. Multiplied times 100 gives me percent, is about 8% of the time I got recombination. So I'm going to use that as my estimator for how far apart they are, which means they're about 8 map units apart. So the little problem I did was 20 map units, right? So this would be about 10% and 10%, but here it's about 4% and 4%, right? Because I've got about 8 map units. So 3 basic things. Most of these problems that you're going to see that deal with this type of linkage, this 2-point mapping, are they linked? What's the configuration? And what's the distance? Somewhere in there, you'll have to answer that. Now, some problems will tell you, hey, they're linked, by the way, and you'll have to figure out the rest of it. Sometimes, whatever, you may only have to figure out two of them, but think about all three of these questions have to be answered either in the problem or by you trying to figure it out yourself.